Hi everyone, this is Miss Longley, and this is part 2 of the series lesson on meiosis. We're actually going to start with the coloring sheet now. So go ahead and turn to your packet, page 7. You're going to see something that looks like this. With this, we need to kind of remind ourselves that the two goals of meiosis, remember, is to make gamete cells that contain half the number of chromosomes. So we're basically going to result in haploid cells. And we also want to make sure that these gametes are genetically unique. So just keep those two goals in mind. So on page 7, what we're going to be focusing on is steps 1 through 3. There will be a third screencast that will cover steps 4 through 6. So here's what you need. You need something to write with. So I'm going to be using a pen. I highly recommend using a pencil because you might draw a chromosome and you're not happy with it. You might want to redo it. So use a pencil. The other item that you're going to need is something to color with. So I'm using Flair pens, pink and blue. You can use whatever colors you want. It might be easier to copy the same colors I'm using just to make it easier on yourself. And the first thing we're going to do is make a key at the very top corner of this coloring sheet. So what this key is telling me is that chromosomes that are colored pink are going to represent DNA that is coming from the maternal side, so your maternal ancestors. And then the blue is going to represent chromosomes that represent the paternal side, so ancestors on the paternal side of your family. So we're going to use those two colors to color our chromosomes, but we're not quite there yet. So let's go ahead and start with step number 1. So we're going to complete the diagram by drawing all of the chromosomes in the cells that are empty. So you may notice that with meiosis, again, this happens twice. There's 2 cell divisions. We have prophase, metaphase, anaphase, telophase. And then it goes through cell division again. So we got prophase, metaphase, anaphase. You'll notice that some of the cells are blank. And we're going to draw the chromosomes here. So this first one on this side, I'll do this one with you. So anaphase, if you remember, is where the chromosomes are going to be pulled apart. So this is actually separating the sister chromatids. So go ahead and copy what I'm doing right here. With these chromosomes, you'll notice that we have a big one, a medium one, and a small one. So I'm going to try to replicate here this in the cell. So there's the big one. There's a medium one. There's a little one. So I'm going to do that again on the same side over here. Okay. And then I'm going to add my spindle fibers. And again, we'll go through the details of what's happening here later. Okay. So there's anaphase. What I want you to do next is looking at this side. I want you to copy exactly what you see. Notice we got a big one, medium, and a small one over on this side. So go ahead and pause the video, go ahead and fill in your chromosomes, and then we'll check back in. All right, hopefully you're able to fill in this side with your chromosomes. It's okay if it's not perfect. Just try your best. Okay, so now that we have our chromosomes all drawn in, now we're going to go ahead and start now with step 2. So it says draw a dot on each centromere. So remember, the centromere is what's holding the sister chromatids together. So in this picture here, I'm going to draw a dot where a centromere would be. So here would be 1, 2, 3, 4, 5, 6. So I see 6 centromeres. I'm going to jump over again to step 3. So next to each cell that we see here, the number of chromosomes, we're going to write that next to it. And we're also going to say if it's a haploid or a diploid cell. So in this cell, I made 6 dots. So that tells me that there are 6 chromosomes. What you're seeing here are 6 replicated chromosomes because this cell has already undergone interphase. So it's made a copy of every single molecule that's DNA, which is what we're seeing here with these sister chromatids. So I made 6. So next to this box here, prophase 1, I'm going to write 6 chromosomes. Now, back to our meiosis overview. Remember, we're eventually trying to get so that the gamete cells have half the number of chromosomes, so haploid, but we're starting off with this parent cell here. So this parent cell is diploid. So I'm going to write a little note. I think what I'm going to do is write it right above these boxes. So this is a diploid cell. Okay, so diploid, this is 2N. Okay, so what you're going to be doing is for every cell that you see, you're going to draw some dots. However many dots you drew, that's going to tell you how many chromosomes there are. And then you're also going to think about, okay, if it's 6, it's diploid. If you get to 3, it's haploid. So let's do this next one together so you can see. All right, so I got 1, 2, 3, 4, 5, 6. So 6 chromosomes. That tells me it's diploid. 2N. Okay, so go ahead and go through the rest of these cells all the way till you get down here. Okay, go ahead and pause the video and then I'll go ahead and show you what I have. All right, so I have gone through and colored in all of my chromosomes. So let's go ahead and look at which ones are diploid and when do they become haploid. Okay, so again, we started off with this parent cell. Let's pretend this is in the testes. So this is a germ cell. It's already gone through interphase, made copies of its DNA. Now it's in prophase. We counted that there are 6 chromosomes, therefore it's diploid. It's going to go on to metaphase. Still, I counted 6 chromosomes, therefore it's diploid. Moves on to anaphase. I counted 6 chromosomes. It's still diploid. And at this point, the cell then divides into 2. So when I made those dots here, notice we now have 3 chromosomes. Therefore, these cells are no longer diploid. They're haploid. They have half the number of chromosomes. So we've now made it to that haploid stage. Remember, the whole goal of meiosis, that first goal, is to contain half the number of chromosomes. So we've now done that. All right, so we still have another cell division to go. So in prophase 2, which we can see right here, still 3 chromosomes each. In metaphase, 3 chromosomes each, haploid. Anaphase, haploid, and you guessed it, telophase cytokinesis 2, haploid. So you can see that each of these gametes are haploid because they only have 3 chromosomes. All right, so that is it with step 3. Go ahead and watch the next screencast, the final part where we will be covering steps 4 through 6. Thanks for watching.